Unfortunately, we're out of time for questions. So the next talk is by Wolfgang Busch from Vienna.
Okay, great. So my lab is interested very generally in a very common question, and that is, how does the genotype of an organism determine its phenotype? And in particular, we are also interested with regard to phenotypes and genotypes that have some relation to adaptation and the environment. And for this, we use Arabidopsis thaliana, which is a small weed and a model organism, but which is also very suitable to ask those kind of questions, how naturally occurring genetic variation shapes phenotypes, and also how they relate to the environment. And as you can see here, this is basically a summary slide of decades of Arabidopsis geneticists' work, who basically collected wild strains all over the northern hemisphere of Arabidopsis, and you can see that every little dot is a different strain from a different point of the world. And as you can see, Arabidopsis has somehow managed to colonize very different climates, ranging from northern Sweden to northern Africa. And so it is this feature that allows us to actually ask questions that relate to the genotype and phenotype and environmental adaptation very well. Another feature is, of course, that several groups have been very successful in sequencing or genotyping those accessions, how we call those strains, with very high resolution. Now, in my lab, we use the root as a model system for this, and I can show you why. That is because roots of different strains that are genotypically different develop very differently, even very early in plant development. And you can appreciate that those 5 different strains actually display very different root development and organismal phenotypes here, while, for instance, the second strain is a very slow grower. Some of those are very fast growers, and you can see that the branching pattern, for instance, is also very interesting. So we can use this as a very good way to quantitatively and accurately determine phenotypes and ask questions about the genotypic basis for this. And for this, we use tools of machine vision to accurately take images like this and quantify organismal phenotypes from Arabidopsis. Now, in this story that I want to present here, and this is just a few of the projects in the lab where we've been successful in identifying genes, their alleles, and even genetic networks that underlie phenotypic variation in the root system. We basically ask a very fundamental question to here, and even if you can see it very nicely on the slide, you might appreciate that starting from a very simple structure very early on in development, roots develop into very complex 3-dimensional structures in the soil. And those distribution of the roots in the soil, as you can see here in a mature Arabidopsis plant, determines the function of the root, how well it can absorb and acquire nutrients and water from the soil. And the big question, and kind of a very important question in the root field and for plant research in general, is how do we actually get from this early stage to the late stage, and what actually determines this complex 3D shape. And something that has come out of a lot of studies over decades of forward genetic screen is that one molecule, Auxin, which is a plant hormone, is essential in regulating many of the aspects that actually are very important to go from this very simple form to this complex form, because it regulates all the processes that are mentioned here, like the growth rate, the root growth orientation, and the branching pattern. But it's really not understood how this actually relates to phenotypes and natural occurring populations, and how it is actually possible that this molecule can do so many things, but still do something specific for root system architecture. Now, we try to address this using our accessions, and we used something like 250 different strains for which we knew the SNP patterns in very high with very high resolution. And here you can see 4 of those different strains and their response to an auxin transport inhibitor. So this auxin, this hormone is transported, and the transport is a very important part of this hormone signaling pathway. And so if we perturb this pathway chemically in all those different lines, we get very different responses. Some of those strains basically are quite resistant to this auxin transport inhibition, while others lose their sense of root growth orientation completely. And so we can, using our machine vision approaches, we can actually quantify this accurately and basically map the deviation of the root from their preferential growth vector, which is basically in the direction of the gravity. So doing then a genome-wide association mapping for this trait, for this offset of root growth direction from the vector of gravity, we could map it to the genome. And as you can see here, the 5 chromosomes of Arabidopsis colored differently. We found a major peak on chromosome 5 that crossed our very conservative multiple testing threshold. And interestingly enough, it was located in the coding sequence of a gene here. But it's known that due to statistical reasons and to issues like allelic heterogeneity, actually the SNP that you map can be offset from the causal SNP. We basically first look at the expression pattern of all those genes in this region to see whether some of those genes might have a relation to the biological process that we were looking at, which would be the control of root growth orientation. And only one gene, interestingly the gene where the SNP was, where the marker SNP was located, and showed a particular pattern that we could relate to the biology, while all the others were more ubiquitously expressed. And the reason why this relates to this trait is that it's expressed in this part of the root, which is the root cap, and in this part of the root, which is the elongation zone. So in the tip of the root, gravity is perceived, and if there's an offset of the root from this vector of gravity, an auxin stream, and you remember that was basically the process that we perturbed during the mapping, is generated that only goes along one side of the root, and that leads then to an asymmetric growth that leads to a different curvature. So basically this part of the root would be a very hot candidate for a gene being able to modulate this auxin stream in order to basically determine the sensitivity of the auxin system to this perturbation of its transport. Now, consistently with that, when we group the accessions in haplogroups, basically groups that show one allele of this exocyst or the other, we actually can see that there are a couple of polymorphisms, if you look at the full genome sequence, that differentiate this one group that is basically that is not very sensitive to this NPA auxin perturbation, and the other group that's very sensitive to this perturbation of auxin transport, including a non-synonymous amino acid SNP that leads to amino acid exchange in the coding region of this gene that's called the EXOCYST 70A3 gene, a terribly long name because it is it's basically a subunit of a very important complex for cell biology that is involved in exocytosis. And interestingly enough, auxin, the transport of auxin has a very important endocytosis and exocytosis component. It's also, on another hand, very interesting because while in all eukaryotes, except for plants, there's one copy, one isoform for each subunit of this exocyst, of this very conserved exocyst complex in land plants, this specific subunit has radiated. And for instance, in Arabidopsis, we have 20 isoforms of this specific subunit, while in yeast or in animals, there's only one. Okay, so, but we didn't want to stop there, of course. We want to basically get more genetic evidence that this is the causal gene. And so we basically looked at different lines. For instance, we constructed CRISPR truncated versions of this line, but we also constructed overexpression versions of this gene. And I'm going to show you data that relates to the overexpression, but the CRISPR data is very similar. And basically, when we tested the effect of the overexpression of this subunit, we actually can see if we if we basically then test how well does the root actually find the vector of gravity, and that's the process that we think is involved in, we can see that this wild type actually turns nicely when we turn it to the side and mess up its growth vector, while this overexpressor actually does respond much more sluggishly. When we do the statistics, we can see that this is actually very different. So we actually get a very different dynamic of this when we perturb the genetics of this gene in terms of like getting actually to reorienting the growth of the root to the vector of gravity, as you can see here. And interestingly enough, when we go back to those strains with those different alleles of this exocyst, this is reflected in those strains. So this is the same measurements for 3 different strains that actually have one version of this exocyst gene that resembles much more this Columbia wild type situation, and the other group of accessions that have the other version of the exocyst actually resemble much more the behavior of this overexpressor. So basically providing evidence that this is really the causal gene. We checked then a lot of other traits in this overexpression line, where we actually have overexpression of this subunit of this exocyst subunit all over the plant, and we only found one trait that was also altered, and that was the lateral root density. So that is basically the branching pattern. So this is altered when we overexpress this gene, and it's also altered in the strains that have different alleles of this exocyst. And so coming back to our initial question, those are actually 2 very important processes that basically lead from this very simple structure of the root early on to this very complex root structure in the soil, and that is how often those roots branch and in which direction they grow. And so we were interested to actually go a step further and see whether this actually has truly a role in forming this complex root system architecture. And so we developed a method where we actually quantified root distribution in the soil. So we grow this Arabidopsis plant in a big pot, and then we chop it with a bandsaw, call this the pot chopping assay, and then we basically use image analysis to assess the distribution of the roots in the soil. So when we do this, we can compare those 2 different haplogroups, one having one allele of the exocyst, the other one having the other, and we can immediately see that the one haplogroup actually is associated with a deeper root system, while this is associated with a shallower root system. And if we only overexpress this exocyst subunit in the Columbia background, we actually get this switch by just overexpressing this subunit of this exocyst subcomplex. So we basically can say this gene is sufficient to actually switch the roots from a shallow configuration to a deep configuration. Now, we can even go a step further because we know which strains have which alleles of this exocyst, and we know where those strains came from. So we can extract the climate parameters and ask, is there any association of the allele distribution to climate parameters? And in fact, the SNP that we basically mapped belongs to the 5% of all Arabidopsis SNPs that correlate mostly with precipitation seasonality, which is essentially a measure of temporal drought. So we did one experiment, which is of course not like a gold standard ecological experiment, but basically tested whether those two haplogroups, so different strains with different alleles of the exocyst, would perform differently under drought conditions. And so we can see here the blue ones, which actually are accessions that contain one allele of the exocyst, and the red ones that contain the other version of the exocyst, perform equally well and not statistically different under well-watered condition. And that is basically an estimate for fitness, the seed number, but they perform very differently under drought condition. So that we think we found a hint that actually this allelic variation of this exocyst subunit that is responsible for the switch of the root system architecture for an organismal trait is related to adaptation to temporal drought. Okay, I didn't talk about another thing. Because we work in a model species Arabidopsis, we basically can do a lot of molecular genetics, so we also basically identified the molecular mechanism, or at least part of the molecular mechanism of the action of this exocyst subunit, and it seems to affect a specific auxin transport protein. But if you want to know more, just talk to me afterwards. So this work was mostly done by Takeko Gua, a very talented postdoc in my lab, with the help of some other people. We collaborated with Magnus Nordberg for getting like really good genome sequences of Arabidopsis accessions. And I thank the funders for funding of the work and take any questions.
We're out of time.
Okay.
Sorry
Great.
about this.
No problem.